Lucky, you can take your seats. Thank you very much. Okay, now let's just move towards our next presentation. We are delighted to have with us Honorable Dr. Ahmad Aslam Khan. He's currently serving as an assistant professor in Department of Bioinformatics and Computational Biology, Virtual University of Pakistan. He has done his PhD from the world-renowned Karolinska Institute, Sweden. His doctoral and postdoctoral research was from Frankfurt University, Germany, Beijing University, China, and University of Chicago, USA. He has 11 publications to his credit, and his research interest includes genome evolution, with special emphasis on evolution of gene regulation. Today, Dr. Ahmad is going to talk on demystifying the genomic signatures in pseudogenes. Thank you very much, sir, for coming today. Please proceed.
Thank you very much, Varda, for such a nice introduction. Where is the control, please? So, as you can see, the title of my talk, Genomic Signatures in Pseudogenes. Before I move forward, I will let you know that it will be most of the time, it will be about the thought processing, which me and my team underwent when we were doing this whole thing. So I will not be going in very technical details of this project. What I will be presenting here will be more how we approach to our hypothesis and how we set the deductions and other stuff. About general structure of gene, a very high school level textbook representation. This is we have a promoter, we have exons, and we have introns. Exons are the coding regions of the gene. Introns are the non-coding region of the gene, and promoter, of course, is the agency which leads to the transcriptional regulation of the gene. Of course, we know that introns are also somehow involved, but it's promoter basically, which is involved in the transcription regulation of the gene. Also, I will like, it's again a very simple thing you already know, but because to know about pseudogenes, it's good that we start from here, that what is the relationship between me as a physical entity and what I inherited from my mom and my father, and that was the information. So it has been around 60 years or more than 60 years, we know that proteins and DNA are interrelated through what we say the central dogma, and you all know about it. We have our gene, it goes through transcription into RNA, splicing comes, and then exons are only there, and then comes the translation giving rise to the protein. We all know about it. And this is in the cellular context, we all know about it. I mean, for me, even if you do hundreds of postdocs, this one figure, this one animation thrills you a lot. I mean, nothing else thrills me that much as this. There is a compartmentalization in one of the most smallest thing, but the sophistication is unbelievable. Unbelievable in whole universe as far as we know now. Until now, we can't see such a machinery. Pseudogenes. Pseudogenes are relic of ancient, I would say, genome. We can say these. Again, if you see this process from starting from the gene leading to the protein, there comes in pseudogenes what happens there. Somehow proteins are not formed in more canonical way. Of course, there are certainly non-canonical types of pseudogenes also, but I'm talking about canonical types. Proteins are not formed, and because proteins are not formed, so somehow we say that these pseudogenes can't perform the function as such. So they are no longer functional. There are three types of pseudogenes: processed pseudogenes. These are the pseudogenes which arise because of the retrotransposition of a gene. Because it's retrotransposed, you all know that it will lose its introns. Not only lose its introns, will lose also its promoter. And because of this, the whole regulation will be sabotaged of that gene, and it can then very easily be pseudogenized. And this is one of the reasons that we see so many processed pseudogenes as compared to other two types of pseudogenes. Because if it is retrotransposed, 90% or more than 90% of the time, it will be pseudogenized. Then there is this duplicated pseudogene. Actually, duplication is one of the most fundamental aspects of all genomes, whether of a mud, whether of a lion, whether of a fish, whether of a wheat plant, whatsoever. But the key thing is here, when duplication occurs, there are many multiple copies of a gene genome. And then there is functional constraint which gets relaxed, because many copies can perform the same function, of course. And then again, as Dr. Mushtaq yesterday talked about Darwin a lot, and this was again from the theory of natural selection, which is always guiding principle for us. It tells that when there are more and more genes which are performing the same function, some of them will get pseudogenized. So this is the duplicated pseudogene. And then finally, there is a third type of pseudogene, which we were very interested. That is unitary pseudogene. Again, if we think about us part of a evolutionary tree, meaning sharing with the ancestor of mice, some ancestor, and of course, the genes of each of us are associated with genes. Sometimes it may happen that a gene A, which is shared between me and my partner, mice, or whatever you say, partner inside that biology. So these genes, one of the genes in any of the species, let's say me, it got pseudogenized. Because of certain relaxation in the constraints, whatever, because every species have response to the logical system, they may some certain circumstances when one of the species gets. Excuse me, and they can hear, isn't it? Everyone else can hear me. Zoom वाले नहीं सुन सकेंगे. Okay. So okay. भाई टीचर जो होते हैं वो हिलजुल कर बात करते हैं. Anyway, so these arrows show. Where is pointer? This is pointer. This arrow shows that somehow some disabling mutation does occur in one of the gene for whatever reason, and it gets pseudogenized. Now, this is very interesting case here. In one species, we see a gene which is functional. That is the functional ortholog. In me, it is non-functional. And it really hit our brains. We thought that these must be very important type of genes. And it struck our mind at that time. And what we thought at that time, is there something special in the genomic signature of unitary pseudogenes? Of course, one is getting pseudogenized, the other is functional. So what could, in other species, what could be the reason? So that was the first question which occurred in our mind. Again, then, if we see the structure of the gene, we thought that perhaps the promoter has something. And you can think why we didn't go towards intron and exons. I leave it up to you guys. Maybe you can ask at another time. But promoters, we thought, must be very important here. They might have those signatures. The metazone genome, the metazones, we all are metazones from, we all multicellular organisms, which are from which all the animals originated, we all are metazones. Metazones have a very peculiar feature in their genome, that their genome, that the CpG are depleted because of the process of methylation. But this is not the case in mammals. In me, in all the other mammals, CpG islands are aggregated in certain part of the gene, that is the promoter part of the gene. They're clustered over there, and we say them. I can't. Sorry. ये इसकी light नहीं जली. Anyway, you can see there that the around the 5' end start TSS transcription start site. There is a in the promoter region there is accumulation of the CPGs, and we say them the CPG islands. Fine. And then from here we made again a sort of a hypothesis that what is the status of CPGs in these unitary pseudogenes? Of course, that was the most natural question which would have come in anyone who is working on this side of things. Then we compared the CPGs present in human UPGs and mouse UPGs, and you can see here that amazingly, very strikingly, you can see the genes which lack CPG islands are much more frequent as compared to genes which have CGIs. Fine. So the UPGs in their promoters lack very significantly lack CGIs. Fine. Okay, we go forward. Then the next question, the most natural question, might be, we thought, lack of CGI might be a causative event. What does this mean? That might be this lack of CGI is leading to the process of pseudogenization. This is what came into our mind. So how can we show this? This is a hypothesis. The lack of CGIs lead to pseudogenization. What deduction you can make it, or what experimental settings, experimental maybe dry lab or whatever, wet lab, it's up to you, because we are doing a dry lab work. So the experimental setting for me was this, that what if I see the CGIs in the functional orthologs of these genes, the UPGs, the functional ortholog. Let's say if in a MAD there are 100 or whatever CGI UPGs and they lack promoters in majority or 90% of these genes, then what if we see the functional ortholog in mouse? The status of CGIs in functional orthologs of mice. So if it's a causal event, the correlation is like that. If it's a causal event, that lack of CGIs leads to the pseudogenization of the genes in me, then we must see the same lack of CGIs in the functional orthologs, that is in mice also. So, and this is what we said. We saw here that, So you can see here the matching of CGIs between us and between the mice in terms of, I mean, in functional orthologs and in UPGs is around 90%. So it can never be consequential event. It has to be causal event, because if the genes which are present in mice, they also lack CGIs, and genes which are present in me, they are pseudogenized and they also lack CGIs. It means that somehow nature is selecting those genes for pseudogenization process which lack CGIs. Hooray. We can say, I mean, of course, it's a bit more, I would say it's not that straightforward. We can say, yes, it is most probably a causal event. And then question came, what about the lack of CGIs in other two classes of pseudogenes? And people already knew it, that the CGIs are not present or lacking very much in the processed pseudogenes and unprocessed pseudogenes. And without even knowing about it, you can intuitively think about it. You can think about it, DPGs, for instance. DPG have duplicated genes which underwent some sort of disabling mutations, and you get a pseudogenized gene. And this pseudogenized gene, then what will happen, that it promoted with the passage of time will ultimately also lose the CGIs, because functional constraint is there, it's a pseudogene. Same is true for PPGs also, even more strikingly true, because not only they are under functional constraint because they have an extra parental copy also, which is functional. Also because of the reason they lose the promoter because of retrotransposition, and they lose the introns. So all the regulatory elements are gone. Of course, there will be no CGIs in them. So CGIs here is a consequential event. Even without reading it anywhere, you can think about it in your mind. So it must be a consequential event. So what will be the deduction out of it? What setting you can think about from here? How can you show that it's a consequential event? As we did this with the UPGs, so we will compare these genes with the functional genes for these genes, functional homologs of these genes, that is of the PPGs and of the DPGs. And again, we saw that now, of course, if it's a consequential event, so what is meant that by consequential event? That after the process of pseudogenization, the gene will lose the CPGs, it will be consequence. It is not leading, so the loss of CGIs is not leading to pseudogenization, it's other way around. And this is what we saw here. And you can very clearly see here that there are, without CPGs, in PPGs in parental type, sorry, this is parent of PPGs, and this is PPGs. In PPGs, you can see a lot of without CPGs, but in parent types, there are very less CPGs. Okay. Then loss and gain. Of course, we wanted to see numerically how many genes were lost, and we could see that in PPGs and DPGs as compared to the parent types, lots of genes were lost. So again, our hypothesis is to some extent we can say, yes, this is most probably the case. It is a consequential event. So in this way, we learned and we showed to the world that pseudogenes don't just throw them away because they are maybe not functional. Maybe they are hinting towards some very, very important features of the genome, history of the genome, stability of the genome, evolution of the genome. We should not ignore these important relics of our genome. And this was then a paper which was published this year in Mammalian Genome, a very nice journal, you all know about it. And then a secondary paper, which was offshoot of this study, we came across a gene. We thought that it is showing some transcription read through, in which one gene is controlled by the last exon of the previous gene, adjacent gene. So we thought that this process also leads to the process of pseudogenization. What is the take-home message here? The take-home message, of course, we had a paper. Of course, we showed to the world that lack of CPGs leads to the pseudogenization. Of course, we showed to the world the lack of CPGs in PPGs and DPGs is entirely different from lack of CPGs in UPGs, in unitary. But even more important lesson for all of us here is beauty and essence of science lies in the curiosity in knowing the unknown and not in what profit it will give to us. इतने पैसे मिलेंगे, इतनी funding शायद मिल जाएगी, इतना impact factor whatsoever. I think much more is the curiosity to learn. And if you have that curiosity, I bet with you, at the end of the day, it will give profit to mankind, to the humankind. And it has been repeatedly shown over the years, over the century, from Einstein from Darwin to Einstein to Abdul Salam, whatever you say, whoever you choose, even Watson-Crick, they never did their experimentation, their studies to get some financial benefits for mankind or for themselves. But at the end of the day, the financial benefits, those theories, those studies gave immense. So this is for all of you who are my students, who are my juniors, even my fellows. This is the beauty of science, to learn about the unknown. of course, then I am very thankful to my department, bioinformatics and computational biology, and of course, my fraternity of life sciences. Most of them are sitting here. And of course, most importantly, my students with whom I did this all. Awais is there, Anis is there, Saima is there, Farah is there, and so on. And some Mahin is also there. Some of them are from Biju, some are from, one is from LUMS and one is from Qaeda University, but most of them are from Biju. And thank you very much. Any question, please?
Excellent, sir. Any questions from anyone? Sir, I will have the honor to present you the mic with my own hands, and you can then take a start with your questions. Just give me a moment.
It's pretty nice to see all those graphs and all other things. I'm just wondering, let's say what we know that at the time of the gene duplication, the two copies of the genes, the daughter genes, or you may say the genes which are coming from the antecedent genes, they are completely identical at the time, nascently they are. So then deciding which one is parent and which one is a daughter, don't you think so? It's a nearly impossible task. So when you say these are parental and these are that, I mean, even the phylogenetic tree can't resolve it. All right. Mm-hmm.
But there is a very good database which is providing you the parental of each process and unprocessed. Parental genes are probably we are not comparing with the two species here. Of course, I have a retrotranspose gene of retrotranspose and present in one chromosome, and the gene A-dash is another chromosome in me. So the parental gene is the one in me which is functional. From which, I mean, let's say there was a proto gene which was ancestral to these two, ancestral method gene A that underwent retrotranspose, it's got retrotranspose, and one became A-dash and one A1 and A1 gets part inside and got so the one which is not transposed.
So let's say, okay, so let's say we got 4 Hox gene cluster.
Yes.
So it's bouncing back the same ball which you yesterday threw to me about whole genome duplication. So you got 4 Hox gene cluster, and there is only one which has been present in all invertebrates, right? In any invertebrates, let's say starting from after the common coelomates, through coelomates. Okay, so like we have Hox A1, Hox B1, Hox C1, and Hox D1. Okay, these are the 4. Hox C1 is a pseudogene, we know that. So, but then we have A1, B1, and C1, D1, which are the intact functional Hox gene. Which one you would consider to be an ancestral?
These are genes which are from the duplication.
Genome duplication, not gene duplication.
These are the duplicated genes.
Yeah.
This is that now for this ancestral, I mean, as I know from the data from where I expected, I don't know which one of these specifically.
No, forget about how will be decided the principle.
I would say the one which shows a maximum in terms of and also one which have in its regulatory region much more again that again comes to which maximum alignment and then they do it by versa. That is the only way then
Okay, Yeah, for me, it's slightly dicey. No, that's fine. You can publish. I am sure this is a very excellent. I completely agree with you. It's a peer-reviewed article. It's just for my. Uh, it's just for my own education. I'm asking you. Yeah. Okay. The other thing is that have you normalized, like you have shown a histogram where you have shown the CPG island which are being present in the primates, in the primates and in the rodents and mice. Have you ever thought of normalizing that with reference to the number of the genes or gene families or the genome size per se? So what my concern is that, so say for example, if you have a genome size of 1,000 base pair, hypothetically speaking, and there is an organism which have a genome size of 2,000 base pair, and we randomly assume that after every 10 base pair or 100 base pair, there will be a CPG island. So in the case of 1,000 base pair genome, you would find 10 CPG island, right? And in the case of a 2,000 base pair genome, you would find on the basis of the random space and time. Now, hold on a second. On the 2,000 base pair genome, you will find 20 CPG island. So if you just discard the genome size, you would say, oh, that particular organism is a higher CPG island compared to the other one. But it is precisely because of the genome size.
Because we didn't have time, because I would have said it more clearly, that it's not about the size of the genome, it's about the number of genes. The CPG islands are aggregated.
Same it goes. The reason being this, rodents are being notorious. They have been diverged from the primates about 92 million years ago. Okay. And after divergence from the primates, once the, what is that, legomorph, the rabbit group actually separated from the rodentia, then they undergoes extensive expansion of many number of the genes.
So, honorable doctors, I'm really sorry to interrupt you both. We took a great deal of time in this, but I would humbly request Dr. Mushtaq to resolve his remaining queries while meeting Dr. Ahmad personally. He will be outside for lunch, of course. So you can ask all your questions from him later on.